My name is Yves Tinevez. I work in a core facility of the Institut Pasteur. So, just as a presentation, I am not a researcher. I'm a research engineer. I do not work in a lab. I work in a research infrastructure. And these are the very courageous and brave people that work with me. Our mission is actually to help researchers, typically in biomedicine or biological imaging, with the techniques of image analysis and bioimage analysis, actually. And I must say that I'm a physicist. I come from the microscopy world, and that's the typical image I'm used to deal with. So this is a section of the epithelium of the colon, and in that case, it's infected by a bacteria in green. And when Perrine and Ayoub invited me to contribute to that, I didn't know how to position myself, right? And so what actually Perrine asked me to do is really to make an introductory talk, something entertaining, but there will be not that much of a scientific content. I will try to motivate the importance of image analysis using projects I have done for the users of my core facility. And so from the outside, I've tried to check what you folks do in imaging, and I noticed that you actually have a quite large scale of range that you want to image and of species, as was said, you go from viruses to whales and so on. But in my case, and in the next 20 minutes or so, I will take example from my work in the Institut Pasteur. We are focused mainly on human infectious diseases, and that's typically what we do and what we was here. For instance, here's a picture of our wonderful and fantastic founder, Louis Pasteur, here in a painting with a beautiful microscope. And this is typically the kind of scales we deal with when we do light imaging in the Institut Pasteur, right? That's the kingdom of light microscopy. That's the range of scales you can access more or less easily with a light microscope. It goes, for hundreds of nanometers to a fraction of a millimeter. In the case of infectious disease, actually, this is very inasmuch because you get bacteria, but you also get the host cells and so on. You miss the structures of viruses, but other technique and other technologies are here from that. So imaging for infectious disease, it's always been a love story. And for instance, here I would like to introduce Robert Koch as a pioneer of this. It's a great competitor of Pasteur. But this fantastic researcher actually made crucial advances in microscopy for imaging diseases and infectious diseases in particular. Even as a medic, he recognized the importance of visualizing the effectors of diseases, that is pathogens, and typically bacteria. He made plenty of technological advances that are quickly described here. And for instance, his lab is credited with the first photographs of bacteria, not only drawings, right? And now if you fast forward 200 years, things have changed quite a lot on technology, but we still use imaging to access infectious diseases. Here, for instance, what you see in green, that's the parasite of malaria. That's one of the greatest human killers. Here, in that case, image in a mouse, and this is just an experiment where we actually bite the mouse on the ear with an infected mosquito. And half an hour, 1 hour later, you find the malaria parasites in the liver image here, right? And imaging gives you, imaging gives you a direct insight of what's happening, what are the dynamic processes of infections at the single cell, single bacteria, or single object level. I think this is unique, right? And this is fostered by these beautiful devices called the microscope. Now, again, that might be a little bit distant from what you do in ANERIS. I still want to take some time to motivate the importance of image analysis via the fields I'm using. These are what we call the big four, all the modern microscopes. There are much more brands that do that, but these are the 4 lens polishers that work in life sciences. And you place a sample here, you shine light through, and you get this kind of image. Now, this is actually a fibroblast, that's one of the cells that runs over your muscles, and image here on a microscope without any special contrast technique. If you want to do the analysis of that, it's going to be a bit tricky because the contrast is very poor. Here you have like non-specificity, and the image content itself is not that interesting. It's a pity for us because most of the animal cells, they are transparent, except when it matters, and that's an issue for actually for analysis. You kind of need to work on different contrasting technique, either staining the sample, either working on the optics. One of the technique among everything that exists that I would like 5 minutes to introduce, is fluorescence microscopy. In the framework of biology, it's fantastic, mainly thanks to the discovery and the ability to clone fluorescent proteins. So I will come to that. And also on the side of microscopy, the development of technology that allows exploiting the properties of fluorescence. So Ayub and Perrine told me to make something very general so that we can address the widest audience as possible, but I guess many of you already know that, right? A fluorophore, it's a chemical that can be anything, but that has a very unique properties. They absorb light in one color and re-emit light in another color. Here I put the case of a GFP, that's the abbreviation for green fluorescent protein. So it's a protein, so you can express it genetically, and it emits green light when it absorbs blue light. There's plenty of chemicals this way in life science, and actually I'll put 4 examples here. For instance, DAPI, it absorbs UV, emits blue. GFP, we just seen it. DS-Red, that's another protein, absorb green, emit orange, and then Alexa fluor, absorb red, emit infrared. What's interesting for us is that all of these, they can be used in imaging technology. For instance, if you want to stain nuclei, DAPI will go into the nuclei and stain them. These are proteins, so you can alter the genomes or actually express these proteins into your protein coupled to the protein of interest. And this is a chemical that you can couple via antibody labeling. I will not go over to that. This is exploited in special microscope design. For instance, in the case of GFP, they are made to shine blue light on the sample and collect only the green light so that you see only the part of the cells that you stained. Here, I have stained the microtubules of a cell, and then this is what I image. I really see the cell itself, the structure, with a very good contrast, something that is specific. I see the microtubule and only them, and something which is sparse. Now, this is important. The majority of pixels are black, and the intensity of light encodes actually the concentration of the protein, right? So this is especially interesting for us, and it fostered actually the interest of people like me coming from physics that are interested into quantitative imaging. Okay, just parenthetically, I showed you an example with one fluorophore, but the optical spectra is wide, and then you can couple many of them. Here, for instance, in a strategy where I have 3 fluorophores, I could stain separately the DNA content of a cell. In green, you have the actin, that's another part of the cytoskeleton. And in red, you have the mitochondria. This is where the cell produces energy. And so you have excellent image of excellent quality, sparse. And also now you have the context, where is the cytoskeleton related to the DNA and so on. And so this fostered actually the field of bioimage analysis because of interesting properties of detectors. First, people observed that the cameras we were using in I think 3 decades ago, or more than that actually, were highly linear compared to the photographic plate that were in the beginning. At the same time, the fluorescence effect that I described was a linear effect as well. And so you could go beyond actually imaging and actually use light microscopy as a way to follow the dynamics of molecular processes. You see? So not only you image the effectors of, for instance, an infectious disease, a pathogens, a host of a pathogens, but you could also follow molecular processes, gene expression, translocation of proteins, and so on, and actually almost have quantitative information about the concentration, for instance. And this fostered the field I worked and all my colleagues work here today, bioimage analysis, something that allows you to make sense of images, what you see, quantitatively. Briefly, so this is the kind of image we did. We've seen you in the introduction, beautiful images of fishes. This is what we deal with nowadays. It's just pictures of a cell. And in the computer, it looks like this actually. The computer doesn't see images. The only thing it sees is actually an array of numbers. In case it's a matrix, right? In our case, if you remember, if you use fluorescence microscopy, the intensity of the pixel encodes the presence of structure of interest. So here, in that case, you see that I have pixels with a large value, and that's because I stain for the DNA, and so I image the nucleus. In the cytoplasm, devoid normally of DNA, I just have background noise, and so the pixel value is low. You can exploit that in what's called a segmentation strategy. That is, you will move from this array of numbers to actually a higher level that describe your image, that is segments. In that case, I was able, using a simple analysis techniques, to partition the image into nuclei and cytoplasm. This is what's called segmentation. It's a huge topic in image analysis, and it's the topic of one full day of this workshop, I think the third one and so on. And there's a huge literature about that. It's super difficult. And we will see some of them in a moment. And so image analysis, if you want to summarize it to the extreme, it's a bit like this. How do you move from this to that? It corresponds to condensation and formation, right? You move to simply a huge number of meaningless numbers to something higher level, but that's the only one with which you can do science. Alors, in the second part of this talk, I think I have still like 15 minutes. I would like just to entertain you with examples of image analysis that what we've been doing in the core facility, just to illustrate, what would be the impact of imaging on science. Most of what I will show is either focused on tools or very preliminary. Hence, there will not be big, conclusions on science and everything. I will mainly present the techniques. Also, because it's just a catalog, I try to order them by size. And so we will start from what I call very big samples on the microscope to very small one. And I will focus, if that's okay, on the main, let's say, interest of the core facility, which is actually tracking the dynamics of cells. Okay, so in the framework of ANERIS, I thought that, I would maybe interest you into a bit of marine biology. And, this is one of my favorite marine biology samples, the shrimp Pariale hawaiiensis. There's a whole field, in biology that's interesting into developmental biology. How do you go from an egg to actually making something fully developed? And here you can image cells, you go to tissues, to organs, then you have a full organisms. And learning how this is made is actually fascinating and also opens the gate to many cellular processes. Now, in that case, to do that, I guess you've noticed, but we are not exactly 2D or flat organisms, right? The beauty of biology goes 3D. And you need to have some kind of imaging capability that can reproduce this 3D. There are many microscope and optical configurations that give you a 3D image. Here, I just put one example. It's the light sheet microscope. The most, probably, if you're into microscopy, you're familiar with spin disk and confocal microscopy. That's the main one. In this case, we use light sheet microscopy. What you see here is the sample here depicted in green. And there's a light sheet that goes through it and illuminate a fine optical section of the sample. And the light is then collected by an objective, and the camera, would be roughly here. And so with this configuration, you capture what we call one Z slice, an optical section through the samples. If you want to build actually a 3D representation of your sample, what you do is that you simply move the sample through the light sheet. You generate a series of Z slices that you can stack just to generate a 3D view. Now, in our case, I insist, this is a true 3D view. That's not a 2D image of a 3D scenes. That's a 3D image where the array of pixels that you have is 3D, right? In the case of this experiment that I'm presenting, it's a bit more complex than that, right? Because this setup allows actually acquiring several angles of the sample to build, let's say, a more accurate view of it, right? And you can do that. You can image the sample over multiple angles, and you can image it over time. And in the end, you get this. It doesn't work anymore. Right. this is this movie. Alors here I'm making a huge shortcut, right? Because building just this movie took a huge amount of time and of techniques I will not survey. But to be honest, I find personally this movie incredible. So here you follow the development of an organism over almost a week at an incredible spatial resolution. This is a full organisms, but we can see and individualize and single out every single cell of the organisms. At later stage, it's a bit difficult, but it's amazing. And we really see all the process. We start from something which is spherical, and you see the rate of cell division is crazy, but then a shape starts to emerge that will be the head. And see here, you have digitations that emerge for this spherical shape. And you can follow that. For me, I'm a physicist, or you move from something which has a spherical symmetry to something which is digitized. I found that incredible. I hope the movie is playing okay in your case, but okay. This is the results. Now, this is all well, this is beautiful. This is a video in PowerPoint. However, on the data drive, the things are maybe less glorious. Generating this movie actually took quite some disk space, right? And the raw data set is 7 terabytes. Again, in the introduction, we've been speaking about the challenge of large data, terabytes of data. Here, the challenge is a bit special because you have one data item, one movie that is 7 terabytes. In our case, we have spent several years to actually develop tools that could simply open the data so that we could visualize it and annotate it even manually. It took us a huge amount of time, right? The movie I just showed was acquired probably 10 years ago, like exceeding the lifespan of a shrimp by a lot. And having the user-friendly tools that allows to browse and investigate this development took us time. But my collaborators and I, that's the lab of Pavel Tomanchak and Anastasios Pavlopoulos, now in Crete, they could use that to actually follow this development and actually backtrack the digitations. In particular, they were able to prove that one of these digitations always emerged from a single row of cells that you see here. These are the stem cells of the digitation that you see. And pushing that, they've seen something incredible, right? One layer of the cells that you see here will give rise to 2 parallel fingers of the shrimp here. Again, for me, I'm a physicist, I find this incredible. In the beginning, this is early stage of the embryo, it's still an egg, and you have something spherical, but the spacing of the cells here, it's on a square lattice, which I find incredible. There's a huge amount of data from this single image that we haven't yet dig, but it will come. The next topic I would like to talk about is now focused on a smaller, let's say, sample that's single cells in the context of cancers. And that's, let's say, a body of work for which we again develop other tools, but this time focusing on something smaller. And in particular, these are good examples to introduce the importance of artificial intelligence techniques and technologies for our field. So nowadays, when you speak about classical image analysis algorithms, people think about non-artificial intelligence algorithms and so on. And the classical algorithms, actually, they are typically very good when you have the images I introduced. Thank you, Marie. For instance, if you have an image that is sparse where objects are bright over a black background, and so they are doing fine. And here I'm just showing an example of detection, right? I want to detect objects, but I'm not interested in their shape. So I have this little magenta balls that you see here, and here and today I have these circles. So this work well and good, but the challenge is, what to do when you have this kind of samples. These are stem cells. Each of the, I forgot to put a scalar, I'm sorry. But each of the dark stuff you see here is a stem cells, drop on a Petri dish, and it will divide over time. And first, it's kind of tricky because contrary to the fluorescence images I showed you, the contrast is not good. The image, there are no salient features that would make it easy for a computer to grab what is a cell. And then the second very hard challenge is that they're very dense. they are touching each other, and they're very hard to disentangle. And then finally, there are some unspecific structure, right? Here I have dirt or piece of oil on the objective that happens that actually stain the image, but are not cells, right? They're spurious structures. And for this, actually, artificial intelligence was like a godsend. I will take now the example of what you see here. It's cancer cells in a wound healing or cell migration assay. My collaborator, Guillaume Jacquemet, in the Turku University, what he does is that he cultivates cancer cells and then you remove automatically a wide chunk of the cells, and then you will see that they migrate north, colonizing the space, right? And this is linked of course, cell migration to cancer aggressiveness. And Guillaume is actually testing different drugs that affect this process. But he does it in an automated manner. And so this is automated microscopy. And what you see here is a plate, a multi-well plate, in which here you have 96 wells, in which you have 96 experiments running in parallel. We have automated microscope nowadays, that can image all of this in parallel and generate, again, a huge amount of data. Here, in that case, the data is big, not because an image is big, but because there are many images in parallel and many experiments in parallel. What it means for us is that the strategy I showed you, where you inspect the data and actually manually backtrack cells, it doesn't work anymore. You have too many experiments in parallel for a human, and by this I mean a PhD, to review all of this manually and create them. You want to have data analysis that is robust without human creation. And here the challenge is that you have cells that are very dense, divide, migrate, and also you have some spurious structures. But we are very fortunate because the people that work in artificial intelligence and at the crossroads of biology have developed fantastic algorithms that can harness this difficulty. I'm showing here the example of StarDist, an algorithm by Martin Weigert and Uwe Schmidt that is very good at segmenting objects that are very close to each other. And just the use of StarDist in our case allowed us to have something very robust for tracking. So I didn't mention here, but you see that we track cells, that is, we propagate the identity of an object through the movie. And this really depends on having a good segmentation, a good and robust segmentation. Objects may not disappear and reappear, it will confuse the algorithm. So what is, this is based on a deep learning network. What is a deep learning network? I'm not the person to ask that. Today you have Estival and Thierry, they will explain what it is much better than me. In the minutes that I have left, it's just based very quickly. It's just based on having an expert manually annotate an image to train a neural network to be able to recognize specific object that it was trained on. So with this process, you produce a pre-trained model, right, that you can apply on novel image, unseen images to segment them automatically. It works, it's much more complex than that, but it works beautifully. And in some cases, you can actually use a pre-trained model on new kind of images, somewhat different from what you train. It's been very successful. Look at this again. The quality of segmentation is really good. We are able to follow cells even when they divide, and the automated tracking results are really good in consequences. And now this study that would be otherwise be brittle and difficult is now kind of not trivial, but much easier thanks to artificial intelligence. Apart from density, these neural networks based on deep learning, they've proved also able to harness like complex samples with very low contrast. Like here you see cancer cells again migrating on a substrate, but this time imaging what we call bright field, the light going through without any contrast techniques. And here we use a well-known deep learning algorithm called CellPose that we retrain. Actually, we manually annotate for these cells. And again, the fiability, the robustness, and the accuracy of the detection is quite good. And so this is, if you start doing bioimage analysis today, what you can find. I'm running out of time. I'm still having 4 minutes. I will try to conclude briefly. just want to show you that, if I connect back to the introduction and telling that we have molecular imaging techniques that allow us to follow the dynamics of a reporter, here's an example, right? These are again your migrating cells, and here Johanna Pilvanainen in the lab of Guillaume Jacquemet, she stained the cells for a molecular reporter called ERK. It's a very wide signaling protein that has a wide range of roles in migration, apoptosis, mitogenesis, etc. And we were wondering if the instantaneous expression of this reporter was correlated to the dynamics of cells. And so once you have the tracks that you see here, you can plug, for one track, the ERK activity, that's the intensity inside the nucleus, and correlate it to the velocity of the cells. And here we find out that there's no obvious correlation. Right. I will actually take the time to speak a little bit, not about cancer, but another fun topic this time, meningitis. Meningitis is a human-specific disease that is caused by a bacteria called Neisseria meningitis. And this is a project we are doing in collaboration with the lab of Guillaume Vinil, just next door, with a very brave PhD student, Laura, here. And so this is what meningitis looks like. So here you have, that's an image by Pierre Nivoire. Here you have in white the human blood vessels, and it's on a murine xenograph. And in green, we have the bacteria. Look at what happens in this very short time. The bacteria comes here, and in a very short time, up, you have your blood vessels completely clogged. And then you have aneurysm, AVC, or that kind of disease, and it's death, right? And it's incredible here. The scale is a kind of quite wide. We don't see single bacteria here, but the force and the speed at which this bacteria grew and the density is extreme, right? It completely blocks huge pressure and so on. And we wanted to study that, right? How these single bacteria, they grow and they can maintain this huge pressure. We don't do that on humans. We have like an experimental setup in which we can replicate the growth conditions, and have them something which is amenable to microscopy. And we have to because the bacteria are very small. These are the bacteria. You can stain them, for instance, for the membrane. And so they look like this, right? In fluorescence, you only see the periphery of the bacteria, or if you stain the cytoplasm, the inert, and you see the volume and so on. And again, you want to segment them, and this time, this is again machine learning, another field of artificial intelligence. You can actually train, again, a computer to actually segment the cells and have, on this image, which are honestly of good quality, good accuracy with that. And so this is what LoRa is, and that's, kind of the resulting movie. When you start, for 4 or 5 bacteria, you see that they grow at a speed which is quite impressive. You can segment them, individual one, and from the segment, you can apply tracking algorithms that will be able to recognize cell divisions and actually identify the clusters or the colonies of the bacteria. And there's a breadth of information in this movie, which I find incredible. Because you have access to the dynamic of growth, you have the mother bacteria that divide successfully. So you have the hierarchy, the lineage of the cells. At the same time, you have the shape of the bacteria. So you have all morphological information. And then finally, you have the spatial position of the cell in the colony. And then you can study whether, the colonies, the bacteria that are on the side die quicker than the one in the center, that kind of things. This is the kind of data you can extract. This is a lineage tree. I will not go into detail, but let's say that, this is time. This is a bacteria. It's been detected over 8 time points. And then here it divides into, and so on. And at the same time, you have morphological information, right? And so you can see a bacteria here, and that will grow and divide. It's kind of difficult to see here, but it seems like that what we see here for the growth of the bacteria is that it seems to elongate. That's fine, because then you can pick one of the bacteria and measure the morphology based on the segmentation. And this is what we observed. That's the area over time for the bacteria. And every time it grows linearly, almost linearly, as you see, and every time there's a division, boom, the area is roughly divided by 2, and this reproduces. And here, this is the circularity, right? That's a scalar number that simply tells you how round is the object. If it's 1, it's a perfect sphere. If it's 0, it's a perfect line. And you see that, just after division, the circularity is quite high, and then it decreases and decreases just before division. And so there seems to be a correlation between that. And this is what Laura investigated. And if you put all these morphological features together, you get that, right? In a plot of cell area versus cell circularity over all moment of the life cell, you see that they cluster into either having low circularity and high area, or high circularity and low area. And this corresponds to actually, in between division and cell division. Again, there's a huge amount of data here, and we're still investigating it. I need to skip this because I'm already over time. I just wanted to, tell you that image analysis, it's the extraction of quantitative information from images, but it's never the final answer. As a bioimage analyst, if you want to go there, you need to see a little bit beyond and know about data analysis, something that can make sense of the image data you've been able to extract. But this is a full topic on biostatistics and bioinformatics. It's impossible to cover that. So this is part of the things we've seen. For the sake of time, I haven't been able to tell you about organ level or tissue level analysis. Also, of urinary tract infections, that also a fun topic, and so on. And we still many of more of what you can harness with microscopy and bioimage analysis. I thank you very much for your attention. These are all the fantastic people I've been working with, and I miss even plenty of them on these slides and so on. Thank you very much for your attention, and if you have questions, I'd be happy to answer them. Thank you.